Thank you very much for coming. Welcome to Biomagune. As you know, we have this year, we have the privilege to host the Christmas lecture. This is the third edition of the Christmas lecture, and the lecture will be presented by Joaquin Castilla, and Jose Maria Mato will take the chair.
So many of you know Joaquin's, but for those of you that may not know his career, I will summarize it. He studied pharmacy at the University of Navarra, and then got a PhD in the working at the University Autonomous of Madrid, but also with the National Center of Biotechnology. And there is where he became interested and introduced to work in virus and infections. Then he was a postdoc for a short period, also at this National Center of Biotechnology. And in the at the same time, he was trying to do two things together, studying medicine and being a postdoc. So he left medicine when he needed to spend too much time at the hospital. And I think this was good for science and for Prions research. So then he moved in 2003 to Serono Research Institute in Switzerland, where he started his second postdoc. And then rapidly, and that's when I think he became interested in Prions, well, just even before this postdoctoral period. And then after some time in Serono Research Institute in Switzerland, he moved to Texas with assistant professor tenure track to the University of Texas. And then moved again in 2006 to the Scripps laboratory that they started in Florida. By then he had really made a very interesting contributions to the Prion research. And we were fortunate that in 2009, he decided to move to Biogune. He applied for one of these Ikerbas research professorships. He got it for the last 2 years, or a little bit more than 2 years. He has been working with us on Prion research. He continues his research there. He has made excellent contributions to the Prion field. But particularly I think it's because he developed a technique that is called protein misfolding cyclic amplification, PMCA, which he will be talking a lot about it. But this has been fundamental to demonstrate that prions are the infection particle. So to the theory of the prion infection, that is one protein, only you need that protein in a special conformation and would be the infection cause. This PMCA has been critical for that. So I think it's a pleasure that you decided to move to Biogune, and we really look forward to your lecture, which will be limited to 45 minutes.
Okay. So thank you very much, Jose Maria and Manuel, for giving the opportunity to be here. I'm really honored to give the Christmas lecture. Today I'm planning to talk about Prions, and the title is saying Minimalist, Pathogenic Agents with Maximum Complexity. Minimalist because probably Prions are the smallest pathogen that we are going to find, that we have now and we are going to find in the future. And maximum because you will see how many things, how many characteristics we have to explain with these minimalist compositions. I'm going to start with a bit of history, starting in 2 centuries from now with the scrapie. This is the first case. I mean, the scrapie is an ovine prion disease, appear in around 200 years ago, and after almost 120 years, Hans Creutzfeldt and Alphonse Jacobs described for the first time the human prion disease, in different patients, and that's the reason why the human prion disease was called Creutzfeldt-Jakob disease. In 1950s, Daniel Gajusek discovered in a very specific area in Papua New Guinea a very weird disease appeared in almost in the 100% of the populations. They were showing clinical symptoms similar where this Creswell and Jacob described before. And the reason why 100% of the population had this kind of disease is likely because one case of Creutzfeldt-Jakob disease appeared in this population, and since they practiced at that time cannibalism, the disease were transmitted to all the population. And in 1957, Gajusek was awarded with Nobel Prize. But the critical person in this prion disease is Stanley Prusiner. Is in 1980 proposed that the reason why we have these three diseases is just a little protein, an infectious protein. And because of this theory, and this is the first time that we knew the word prion. And because of this theory that at that time were not confirmed, in 1987 was also awarded with Nobel Prize. And that time also we observed first case of mad cow disease, that this disease started more popular, much more popular, thanks to this disease, or bovine disease that appeared in Europe, affecting more than 300,000 cows. In 1996, even this disease got more even popularity because of the first case of variant CAD, that is basically a variant of this disease coming from the inoculation of BSC, the mad cow disease, in humans. But there are very interesting opinion about the origin of these diseases, because I told you that it's more or less is around 200 years, the first case of scrapie described, but there are a science publication saying, talking about the scrapie, maybe the scrapie appears also in ancient China 2,000 years before. And this is because of these Chinese symbols indicating that the meaning of this symbol is predators. And these symbols can separate it in two symbols, disease and sheep. And that's the reason why there are people thinking that the first cases of prion diseases in sheep appear 2,000 years ago. So before the concept of the protein, the infectious proteins, the people in these times thought that because they didn't have any other option to think, because the most common microbes were virus, bacterials, all the people thought that the disease was, the responsible of the disease was kind of virus, very slow viruses. In 1980s, Prusiner established the protein-only hypothesis and removing completely this idea. However, during these years, many people, many research groups have fought against this theory because it's really difficult to understand how this little protein can be responsible of everything in prion diseases. That's the reason why I'm going to describe in this talk what basically we have done in this period of time, trying to understand how this protein, instead to be just a little protein, is going to be an aggregate protein in different forms, and it's going to be, and how we can understand all the principal characteristics in this disease with these just conformations. But we are going to start talking about the prion diseases. Starting in the Christmas environment, which species can be affected? Can be affected humans, different ages, different genders, with several different diseases in humans. Creutzfeldt-Jakob disease, as I told you before, but also we have, this is basically 90% of this disease is spontaneous. No mutation in any place, just spontaneous. Nobody knows anything about the origin of this disease. Just appear, and appear most of the cases in people around 60s. But we have some case, very young case, and unfortunately we have a unique case in Spain that this is still alive. It's a girl with 11 11 years. So this is a very unusual, but can be possible. And then we have the same human disease or similar disease with mutation in one specific gene that I'm going to describe later. We have in Basque Country many families with this mutation, so we have at least 3 times more cases with the median all over the world. But as I told you, we have also a classical scrapie in sheep, but also we have, this is a typical infectious disease that appear because of transmission between individuals. But we have also a typical scrapie that appear now, basically are everywhere, and theoretically are spontaneous cases in sheep. In bovine, we have classical and atypical, so this is the typical mad cow disease, and these are the spontaneous cases in cow. The numbers that we are managing is basically 1 per million cases in human and around 4 cases per million in other species. But also we have a very important disease in deer or in cervids, also in reindeers, the chronic wasting disease. This is a very important disease in the US. There are some states with more than 35% of the wild deers affected with this disease. Fortunately, the reason why it's not even more popular is because CWD does not infect human. And we will discuss this later to understand why. And then there are another species like rabbit that are in orange, no, in red, because there is no clear if it's infectious or not. At least we're not clear since 30 years to now. And we are going to clarify if at the end we can see if they can be infected or not. But what we know is that the avians, because they are not mammals, even if they have the same protein I'm going to discuss later, theoretically they cannot be infected. So, but how the process starts in a prion disease? We can start from the gene. This gene is around 20 kilobases, is with 3 exons that everybody has in his any human and any mammal has the same similar gene. Is in an open reading frame is codifying a protein that is synthesized, and we are going to call PRPC from cellular, or for familiarized, it's a good protein, it's a healthy protein. For any reason, this protein is converted to a bad protein, a healthy or pathogenic protein, or pathogenic or disease protein. The most important thing is that we have to know that the differences between this protein and this protein is just the state of how this protein is in solution. This is completely soluble, and this protein is completely insoluble and is aggregated. And this is the key question for understanding everything in prion disease. If for this conversion can happen in some way, there is no any clear explanation how this process is occurring. There's just 2 models. The template direct refolding, where we have 2 different proteins, the pathogenic and the good one, no relationship between them, but one of them, the bad protein, is able to bind the good protein and convert it into in another bad protein, and this process can be repeated. And the other model, that maybe the explanation, the final explanation would be in a mix of both, is a nucleic polymerizations. We have the same 2 proteins, but in this case we have like an equilibrium between them, favoring, of course, in the good protein. The small protein that naturally is produced is being aggregated. So everybody here is theoretically aggregating in this form the bad protein. And this is a very slow process, very, very slow process in forming this nucleus. But if the number of aggregates or bad protein increase, instead to have just nucleus, we are going to have aggregates, small aggregates, and this process is going to be more faster and faster. And this process can happen in 10 years or can happen in 100 years. So theoretically, this is indicating that if we survive 200 years, everybody is going to have these kind of diseases. Fortunately, we will find the solution before. So how we can distinguish these two proteins if they are identical? So theoretically, no difference, practically no difference between these two proteins in amino acids. So how we can distinguish them? So we have a protein, 240 amino acids, with 2 glycosylation sites, soluble form, that in a Western blot we see 3 bands, the unglycosylated one, monoglycosylated, and deglycosylated one. When we digest this protein, the protease destroy completely the protein, and we don't see anything, any signal in the Western blot. If for any reason we have a conversion, we have the same protein, no differences in terms of size or type of amino acid, just the only difference is how this protein is in solution. It's completely aggregated. But how we are going to distinguish in the Western blot that we are going to denature the protein? No difference at all. So this is undistinguishable if we don't do anything else. That's the reason why we have to digest the sample, and instead to have the completely restriction, we are going to just remove a little bit, just a part of the protein, and we are going to have a shift. And this is what how the Western blot is saying us that we have prions in this sample. So if we have 2 proteins that are identicals, what is the principal differences between them? So this is in a structure, just generally, this good protein is based on alpha helix, the bad protein is based on beta sheets, high percent of these structures. Soluble and insoluble, but also this kind of a structure is going to give the standard resistance to physical and chemical actions. But for the bad protein, it's going to be much more resistant. So you can boil this protein and it's going to be maintained the conformation without any problem. So these differences are really important, but the most important difference is that we are going to have the bad protein is going to be infectious, and the good protein is not, because it's basically the protein that we have in our brain and in another tissues. So what we wanted to demonstrate in these years is, assuming that the protein-only hypothesis is correct, that is something that there are many people 5 years ago there, half of the scientists in the field didn't believe in the protein-only hypothesis. So we have to deal in this situation. And we wanted to answer this question: which is the infectious particle? How an infectious protein can show different strains. I'm going to explain you later the meaning of this very important concept, strain. And who is controlling the transmission barrier, something that also is critical for understanding prions. So let's start with the strain phenomenon. The strain phenomenon is a very general concept for many pathogens, especially in virus. Everybody understands that you can have HIV type 1, HIV type 2, different strains, different in the type of proteins in the surface, in the envelope. But how you can explain a strain if you have just one protein? So the first thing that we have to understand that is for us, strains means different behaviors and different practical behaviors. We have, for example, 2 mouse prion strains, strain A and strain B. They are going to have completely different biochemical characteristics. But the most important, they are going to also have different biological characteristics. So if we inoculate a strain A in a mouse, we are going to observe different clinical signs, different incubation period, and different lesion pattern. And if we run a Western blot, we are going to observe different glycosylation profile, electrophoretic mobility, and protease resistance. And this is an example of 5 different mouse strains where we can recognize them just because of this pattern, and of course because these biological characteristics. And in all cases, we are dealing with the same mouse PRP or mouse protein. Transmission barrier. So this is what explains how, or theoretically would explain how a mad cow disease is infectious in humans, but scrapie from sheep is not. And many people here probably have eaten infected brains from from sheep. At least now is not possible because it's removed from the carcass. But in the past, this kind of ill sheep or sick animals were used also for taking the brain, and we know now positively that cannot infect humans. So what is the difference between these two prions, that one can infect humans and others not? But also this is a very basic transmission barrier concept, talking about a species, two different species, very clear transmission barrier. But also this kind of transmission barrier can be shown in the same kind of species. You can have 3 or 4, or even we have even more than 20 different polymorphisms in the protein, all of them in sheep, where compared with the same polymorphisms, you don't have any barrier. But depending on the polymorphism that you observe in different individuals, you can have different kind of transmission barrier. And this means different incubation time, and in some case, even some polymorphisms is completely protective. And in humans, for example, and we have 3 different situations. In this room, we have 35% of people is going to have methionine in the position 129 in the PRP, in the protein that relevant in these diseases. 55% is going to be methionine and valine, and just 10% valine. So why this is important? It's important because mad cow disease is affecting just methionine. So we have around 250 cases in the world of this variant CJD as a consequence of mad cow disease inoculation or natural inoculation, and just all cases were methionine. So this polymorphism is critical for being more resistant to some specific prion. So how we are going to solve this conundrum, which is the infectious particle, how an infectious protein can show different strains, who is controlling the transmission barrier, in the context that we are believing at that time, and we really believe now, but at that time we also believed in protein-only hypothesis, with the protein misfolding cyclic amplification that was designed for the first time by Claudio Soto around 10 years ago, and automatized by the group, and when I was with him in Texas. And this technique, that is basically the idea is to amplify prions, is based on the inoculation in vitro, that's in a tube, of brain homogeneate in these green balls with inoculum, leave incubation, and the units grow, modify. So the bad protein in gray is going to modify, is going to convert the green balls, the good proteins. At that time, we had to differentiate the new generated in vitro molecule, because we didn't know if this molecule, the gray one, the original one, is exactly the same that we were generating in vitro. And that's the reason why. So we have to change the color. This growing had a limitation, likely because of the size of the aggregates. That's the reason why we have to introduce sonication. That's the key process of the method, in order to break these units and help for being seed for new conversions until this process can be repeated, until we don't have more good protein to be converted. From the practical point of view, we have no signal here because the amount of the inoculum is very low, and after the amplification we have the standard 3 bands. So the first question that we want to address was, okay, what we had in the tube is something more infectious, or we have just more signal? Because it's very simple to see the increase of the signal, the bands, 3 bands, but we had more infectivity. So that's the reason why we started a very long experiments, doing 20 rounds of PMCA by one, following with each round with 1 to 10 dilution. The reason why we gave 20 rounds and 1 to 10 dilution was to maintain the amplification in each round of PMCA and to remove the inoculum from the beginning. So the key was to have a new material, newest in vitro material generated in the tube to be compared with the material that we use. And then these two materials were compared using the standard characteristics process, the standard characteristics in a prion disease: electrophoretic pattern, insolubility, protein cadence resistance, deglycosylation studies, structure studies, and of course the infectivity that was tested in vivo in hamster. This is the hamster prion control that we use in the tube as for inoculation in the tube, and all the animals die around 90 days, and the product obtained after 20 rounds of PMCA. 20 rounds of PMCA means around 2 months sonicating, because each round is around 2 days. In each round we have to dilute 1 to 10 in order to remove the inoculum, and after 2 months of sonication we obtain the same infectivity. 100% of the animals die in around 90 days. So prion infectivity can be replicated in vitro and correlates with the level of the signal observed after amplification. The signal that we observed in the Western blot is saying also how the infectious is going to be in animals. So what happened with strains? So if we had a strain A, B, C, D, E, what happened if we use it in the same technique in a tube? We are going to maintain the strain A, B, C, D, or we are going to have a general, in this case, mouse prion disease in the tube. And that's the reason why we did this experiment, starting with 5 different strains, very well known in mouse and very well pretty well distinguishable. We gave 20 rounds, 2 months of sonication, and we took these samples and were compared with the first inoculum sample. And we compared typical parameters like infectivity, like incubation times and other biochemical parameters, observing that prion strain properties can be replicated in vitro. That's a very important step, because at that moment we were pretty sure that the same kind of replication that we observe in the tube is where the replication that we observe in the animal. So the next step was the transmission barrier studies, how we can deal with this important process, phenomenon. This is the first experiment trying to understand if we could replicate the infectivity, hamster in hamster. But what happens if we start with a mouse prion, and instead to use a mouse different, and we use the same hamster substrate? What we are going to observe here? That's the reason why, following the similar procedure, using a mouse, this is a mouse prion using a specific antibody able to recognize just mouse protein. After just 1 round of PMCA and 1,5 dilution, we observe a band with a specific antibody against hamster. So in just 48 hours, we have converted something that was mouse in something that was hamster. In order to verify if this material was really infectious in hamster, we have to give 20 rounds of PMCA in order to remove the inoculum from the beginning. And we compare this with, this is a hamster, standard hamster prion, and with other prions that we are going to show you here. And this is the critical experiment in infectious infectivity experiments in hamster. When we use mouse prions, since we have this kind of transmission barrier, none of the animals, none of the hamsters die after 300 days. You have to wait more than 600 days, and just 50% of the hamsters die with a mouse prion. But what happened with this kind of mouse prion that was replicated in vitro in a tube? Killed all the animals around 180 days, and if we compare with other 3 standard hamster prions, we have different incubation times. And intermediate, we had also intermediate, if we compare the histopathologically samples we have something intermediate. This experiment was done before in animals. A mouse prion was inoculated in hamster, and the results of this infection after 2 years was inoculated again in hamster and again in hamster, and after around 3 years they conclude that they had generated a new strain. So that's really important in prion field, that as soon you are changing the host, the kind of species that you are infecting, you can create new species. And remember, around 20 years ago, nobody knew mad cow disease, so this is likely a new disease generated in Europe. So this is from mouse to hamster, but just for summarize what we can do in prions, in talking about transmission barrier in vitro, this is another example in the other direction. Hamster prion to mouse prions. When we have a hamster prion and we inoculate a mouse, we have to wait around 600 days, and not all the animals are going to die. If we replicate in vitro using hamster prions, we are going to have the same material than before, and again, we don't have huge infectivity because it's still hamster. It's not working. But if we change the substrate in the middle, and instead to use a Syrian hamster, we use a Chinese hamster. This is not a mouse, this is a Chinese hamster. That has an intermediate sequence between mouse and hamster. We have when we inoculate in mouse, we have an intermediate results. So it's like we have increased infectivity because we have now a prion that is more similar to mouse. But if we do the right experiment using mouse as a substrate, now we have the minimum time that we can have, and 100% of the animals are going to die. This is basically how we can deal in the tube, in the transmission barrier. And from the practical point of view, this is a very interesting experiment showing how we can use this technique and have answer in just 24 hours. We have 2 cases in sheep, two cases of prion disease in sheep. It's standard scrapie and BSE in sheep. Most of our sheep were fed with contaminated BSE, mad cow disease contaminated food. And that's the reason why there are cases of BSE in sheep. So the point is, if we cannot distinguish these 2 cases, because for us its clinical signs are really similar and distinguishable, how is going to be the infectivity in humans? As I told you, scrapie fortunately is not infectious in human, and how we can prove that? If we use a human brain in a tube and gave just 1 round of PMCA, we don't have any signal of prions using specific antibodies against humans. So that's the reason why we can confirm that scrapie cannot infect humans. But what happened with this sheep that is showing something identical and in terms of clinical signs to this case, and we do the same. In this case, we have a specific human prion. We have generated in 24 hours the first human prions coming from BSE in sheep. And this is very important because if we know positively that we have in our fields BSE in sheep, in our sheep, we have to change our process because maybe we don't care that we have many cases in Spain, in Europe of scrapie, but now we have to be very cautious if we have many cases of BSE in sheep because it's going to be infectious in human. So in vitro transmission barrier phenomenon can be mimicked in vitro. But I don't want to scare you, but I want to go forward and I'm going to show you how we can do in vitro. Mouse cannot infect humans. That's the reason why we can deal with this disease, mouse disease, in a P2 facility. But we can convert a mouse prion in a hamster prion, and a hamster prion in a sheep prion, and a sheep prion in a cow prion, and a cow prion in a human prion. So basically, depending on what we are introducing in the middle of these in vitro conversions, we can generate any kind of prion starting with any kind of prion species. So in this summarize, so we can replicate in prion infectivity, we can replicate or we can mimic also prion strain properties, and we can deal with the transmission barrier. But who is controlling? So now we have the perfect method that is really mimicking what is happening in vivo, but who is controlling the transmission barrier? So for that, in order to give some idea about what is happening, we wanted to go to nature and selected these purportedly resistant species. These two species that everybody knows that are really well related. And that's the reason. The reason why they are considered as resistant species is because there are some experimental challenge done in rabbits and in dogs were completely negative after 2, 3, 4 years. There are several experiments. But remember that the lifespan of these animals is much longer, and the incubation time of these diseases are really long. We have also information from the shoe experience. BSE was introduced in the food in many zoos, and most of the species in zoos die. I'm talking about felines like lions, cheetah, and other kind of bovine species, but never were found any rabbit or derived species dying because of this disease, or no canines affected with this disease. And of course, no cases in nature, and that's very important. But if we go back to 1990, for example, they were not cases in cow. And then we have 300,000 after 5 years later, we have 300,000 cases. So that's no really important reason why we have to believe that rabbit or canines are not resistant. What happened in our system that we know that is really efficient replicating prions? That's the reason why we're starting with rabbits and using different prions from mouse, from cervids, or from cow. We obtain in vitro the first rabbit prions. Very clear and pretty easy in our hands. So this is against what the people was thinking in the previous 40 years. And in fact, there are more than 50 publications trying to explain why rabbits were really infectious, were really resistant. And this is indicating that rabbits are not resistant. That's the reason why this was really criticized, how the techniques is not so accurate. So what we have to demonstrate if this material is really infectious. Let's see later. What happened with dogs? We similarly try to infect or to convert different prion species, and after more than 2 years working with more than 30,000 reactions, everything was negative in vitro. But as I told you, we can deal with this prion just changing the host in the middle. So we can manipulate or we can direct our prions in different directions until go to the goal. And that's the reason why we choose cats. The reason why we have chosen cat is because when we compare the proteins, the PRP with dogs are really, really similar. And we knew also that cats can be infected in zoos. We have lions, cheetah, leopards also infected. And we have also cats infected with BSE. So that's the reason why first we generate easily, starting with BSE, the first feline spongiform encephalopathy in vitro. And we use this material in order to go to dogs. And that's the reason why we generate also for the first time, the dog prions. But also after modifying our technique, we could also obtain it directly. So the question was, okay, we can generate this in vitro, so there are no limitations theoretically in vitro, at least in mammals. In fact, we have also dolphin prions, we have any kind of prions that or squirrel prions, any kind of mammals can be theoretically converted. But what happened with the infectivity of this material in the homologous species? we inoculate these rabbits in a very long experiment. This was done in Morton using 2 different strains obtained in vitro in 3 animals, because this is a very expensive experiment performed in a P3 facility. And just to confirm that in our hands the mouse prions cannot infect rabbits, this is exactly the same experiments performed 40 years before, and basically the experiments which create this idea about the resistance of rabbits. So after almost 4 years, none of the animals die, even with this in vitro generated inoculant that we saw that those very clearly a prion, rabbit prion disease, rabbit prion. But in one case, in this particular strain, and in this other, in this strain, around 700 to 800 days post-inoculation, we have animals dying. This was studied biochemically, and we observed the typical 3 bands. No doubt about we have the first in vivo rabbit prion, or in vivo you can call mad rabbit disease. And this was a very well studied also by immunohistopathologically, and this is the typical detail, the typical characteristics in a prion disease. The only atypical is the species that we are using, but this is very clear. Nobody is going to doubt about these animals die because of the prion disease. We tried to publish this, but was not enough, because after 4 years saying the opposite, it's not, you have to do even more things. And one of the characteristics in prions is that you have to prove that it's transmissible. Not just that you have the disease, in just 1 case out of 3, we have to demonstrate this infect is transmissible to other individual. And that's the reason why we have to inoculate again in another very expensive experiment, another 12 animals with different amount of this prion, rabbit prion. And at that time, we also generated the first rabbit, the transgenic animal, transgenic mouse, overexpressing rabbit protein in order to accelerate the process, because we didn't know exactly how long it's going to take this process. After 400, this is ongoing, so it's 440 days post-inoculation. We have just 1 case, it's highly positive, so it's 1.5 years. But fortunately, all the transgenic animals, since they are overexpressing the protein, everything is gonna be faster. All of them die in around 250 to 300 days. So this is a definitive proof that rabbits are not resistant to prion disease, but also are really match very well with the previous idea. They are not so susceptible to the standard species. But what happened with dogs? We have this in vitro generated dog prion, and since we cannot use dogs in any experiment, it's really difficult to use them, and it's very expensive and not very ethical in many points. We have to generate transgenic animals overexpressing dogs PRP. Unfortunately, I cannot tell you the result because these experiments are ongoing, but we can tell you that after 200 days, none of the animals have been infected. So what we are learning from these 2 species, why we are using these 2 species, what is the relationship, why this is so so susceptible and this is so resistant. If we go to the PRP, this is a comparative from different dogs, different canines, and a cat. There are just 3 differences, 3 substitutions in the gene, in the PRP gene. So for several reasons, we decided that the key amino acid was this one, because these amino acids appear in other species that were susceptible. So, and this was unique in canines. And when we went to the NMR surface churches, and this is dogs, and we compare with cats, we observe a clear differences in the charges in the surface. And that's the reason why we decided to go to this particular substitution, and we generate a rabbit or a mouse overexpressing this particular substitution. So in this case, we had animals, mouse, with this unique change. So it's not mouse overexpressing dogs. It's mouse overexpressing a mouse protein with that particular change. We had two different lines with 2x of expression, or 4x. That is very important because we should expect that when you have more expression in the PRP, you have low incubation times. If we inoculate the wild type with these levels, we are going to have the disease in 60, 90 days. What we did was inoculate the wild type, that this is the case, with different strains, 4 different strains, and all the animals die wild type after 160 days. If we have these animals were wild type, so we should consider 1x of expression, and in our case we have 2x and 4x. So we should expect, if the mutation is not doing anything, we should expect a shortened incubation time. So this is the animals that were inoculated with 2x and 4x. Fortunately, after 425 days, none of the animals are being shown any kind of clinical sign, indicating that this particular mutation or substitution is a very important, is having a very important role in the transmissibility of the most common prions. And this information is going to be as we are going to finish in, probably we will wait until 600 days, but the idea is to establish new therapeutic approaches based a rational way using this area. Therapies based on dominant negative protein, because this protein with this particular mutation, that is the most important thing, is this natural mutation appear in a very well-known species, can also use as inhibitor of the reaction. For example, we can introduce this protein in a brain, an animal that has been infected, and block the replication as a dominant negative protein do. But also we can do therapy. We can start with therapies based on chemical components designed by docking in a druggable area. This is a druggable area that we are going to start trying to study. We are starting studying for design components. So, starting with a minimalist pathogen with a maximum complexity, and still we have so many questions to be addressed. So this question now, it's amazing how we have studied so many years, so many groups, and at the end we have basically the same question than before. So how do prion cells propagate? Nobody knows. Which is the pathological toxic molecule? Nobody knows. Which are the mechanisms involved in the transmission barrier between species? This is basically what we are doing now, trying to understand much better. We have worked with just one mutation, so we have thousands of them. How we can understand one protein and many strains? This is a question of organization, different conformation. The problem is nobody knows about the structure of a prion infectious particle. So these questions are basically the most important question in any kind of disease. So how we are in this stage. So what I can tell you is that these questions are going to be answered in the future, because now we have in the position to replicate prions using recombinant proteins. And that's the reason why I'm going to show you the first recombinant protein that we have generated in the lab. This is a protein that has been proved is infectious. And as you can imagine now, instead of using brains as substrate in the tube, we can use any kind of protein generated from bacterial and any kind of modification to understand how the transmission barrier works, how we can also label these proteins to visualize them in electronic and in the microscope. We have do basically everything, and also we can produce so amount of protein that can be used in structural studies that before were almost impossible. And of course, this work is, I have to thank to people from my lab and also from other labs, other collaborators, and of course the fans. Thank you very much.
Time for some questions, if anybody is interested. If you have any question.
What is the benignant role of the prions? Why do we have prions?
Yeah, the function of PRP is, I can tell you, it's unknown. There are many theories, there are many publications trying to explain the function, but there are no any clear role for this protein. In my opinion, the best experiment was done by Aguzzi, trying to observe. They observed that the lack of this protein is producing demyelinization, and so that the protein could have some kind of role in myelinization. And this process appears at the late stage, and that's the reason why the knockouts for this protein they are not showing any kind of clinical sign, any problem. So just at the end of around 600 days, they are showing some deficiency.
I see that there is still a lot of discussion about the structure of the infectious particle. Can you comment on the role of lipids in the infectious particle of prions?
Yes, that's a very important question now, especially because since now we can dissect the process in vitro and we can separate what is important and what not. Now we know that lipids are really important in the replication process, at least in vitro. In vivo should be also important. There's some nice publication where the gangliosides could be involved, but these studies again has been done in vitro. And nobody knows exactly how lipids interact with PRP, but everybody knows that some kind of interaction is important. And so this is basically something really new, and we are really interested in that, and we are working on that, yeah.
I don't know anything about prions, so this is a very general question. What makes this protein so special? I mean, the question is it possible that you have other proteins that can be converted like this protein into a pathogenic form?
Yes, the change from something that, so that's, you have, yeah, the point is pathogenic or not pathogenic, that's completely, this is the key, the key word. So you can take any kind of protein, even if the most globular protein, and force in vitro, you can generate something that is aggregated. So you can modify the conversion. That's very clear. The point is how the next step is to force the self-replication for these proteins. And that's something that we don't know exactly what is doing this magic self-replicating process to this. So I don't have the answer how prions are really prions. So what is the difference between Alzheimer, for example, where we have the amyloid beta that also when is able to convert other amyloids, other proteins, and increase the amount of amyloids in a similar way that prions do. But do you need something else to generate pathogenic, to be pathogenic? Do you need something else to really be to be able to self-replicate? We don't know.
I was wondering about the importance of the glycosylation here. Could it be that the prion is a lectin, or that has a carbohydrate binding domain that is exposed or formed and binds to a specific glycosylation?
There are many studies trying to give some kind of role of the glycans. I'm not very fan of the glycans, and especially now that we are basically focused on recombinant proteins, where we are, so we don't have.
You should have not say that here.
Yeah. But yeah, what I can tell you is, yeah, it has to have some kind of role, yeah. But I can tell you that you can generate transgenic animals with this particular, with the PRP, with this mutation where it cannot glycosylate, this protein cannot be glycosylated, and you can infect the animals, so you have a standard prion disease. So, yeah, there are many studies trying to give this kind of roles, binding other proteins, lectins, but this protein is really sticky, so what is true, what is an artifact is difficult.
GPI anchored proteins, is it?
Yes. Yeah.
GPI anchored. Could you comment a little bit of the role of the anchor? Or?
Yeah, that's the other important question. Yes, the GPI anchor is if we generate transgenic animals without GPI, expressing PRP without GPI, we are going to generate transgenic animals where showing this, having this protein out of the membrane. These animals can be infected very well, and they produce tons of infectious prions. So this is saying that GPI is not having any role in prion disease. In vitro, what we have observed that maybe that the GPI has a very important role, because GPI is going to place the PRP in a context to favoring the contact to other proteins and favoring the conversion. So what is happening basically is the GPI is helping just to organize the proteins, but just it's probably a physical role, nothing else. This is our in principle.
Yeah. Another glycan question?
I will make a question which is not my field, so thank you for the presentation. About the discrimination, about good and bad protein, is our body able to give an immune response to the bad proteins?
No, not at all. No, that's another problem with prion disease, because we cannot use any we cannot detect any antibodies at all.
You mentioned that as we age, if we age long enough, we will generate the bad form. Now, if you get samples of people which are very old, 100 years old or so, even if they don't have symptoms of prion disease, can you measure the levels of prions in brain or in blood? And is increasing with age, even if he has no clinical symptoms?
There are no any specific study trying to evaluate if just taking these very old people. But I can tell you that there are some cases of Alzheimer that when we're checked for confirming Alzheimer, they found also prion disease, and in very old people. But I don't see. I don't think there are this specific study just.
What about autopsies or brain autopsies of very old people without symptoms of neurological disease? Those are difficult to get, I guess.
Yeah, the problem is the amount of the samples that you are going to have. In order to, you need thousands of samples in order to have some good correlation. Yeah.
Well, if there are no more questions now, we thank you, Joaquin.
Thank you.